Dear children, in this module, we are going to learn about how the genes and proteins are related. The relationship between genes and proteins are better understood by mutation studies. We know what are mutations. We had learned what are mutations in the previous chapter. They are sudden changes in the sequences of the DNA. So when there is a mutation taking place, there will be a direct change in the sequence of the amino acids which is produced, and thus the functional property of the proteins also changes. So either the mutations which is causing deletions or rearrangements in a DNA can bring about a loss or a gain of a gene, and so also there can be a change in the function of the gene. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion, and such mutations are called as frame shift mutations, or they are called as deletion mutations. Insertion or deletion of one or two bases are called as frame shift mutations. Now let us take the example of this gene. Okay, we can see these codons, codon 1, 2, 3, 4, 5, 6, 7, 8, 9. These are the codons in which this is the initiator codon and this is the terminator codon or stop codon. In the above sequence, we can see insertion of a single base, that is G, has been inserted between U and A. And what happens? The frame of reading changes. Here it was CGU, ACG, UAC. Whereas when there was insertion of a single base guanine, the shift in the reading frame takes place. We can see the shift is GAC, GUA, CGU, and so on. So from that point of insertion, the sequence of amino acid also changed due to the change in the codons. Now in this sequence, we can see there is a deletion. What has been deleted? Adenine has been deleted, and here also we can see the reading frame has been changed from the point of deletion. So deletion or insertion of one or two bases can change the reading frame from the point of insertion, from where it has been inserted or where it has been deleted, and such mutations are called as deletion mutations, or it is called as frame shift mutations. Whereas insertion or deletion are three or multiples of three bases. What will happen if such three bases are inserted or deleted, or multiples of three are inserted or deleted? It inserts or deletes one or more codons, because we know codon is a triplet. It is a triplet. The reading frame remains unaltered from the point onwards. Hence, one or multiple amino acids are either inserted or deleted. In this, we can see this is the normal sequence, in which in the above sequence an additional codon GAG has been inserted, and we can see from that point of insertion of GAG only that particular amino acid has been added, but before that point of insertion and after that point of insertion there is no change in the sequence of amino acids. Whereas in the bottom sequence we can see that ACG has been deleted, and we can see the reading frame remains unaltered from that point of deletion of three multiple bases. Is it clear? So that's a difference between insertion and deletion of one or two bases and insertion or deletion of three and multiples of three bases. Thus, dear children, we are able to understand that if there is a change in the sequence of the genes, there is a direct change in the sequence of amino acids. And if there is a change in amino acids, there will be surely a change in the proteins. And the function of the protein also changes. Now let us learn about the types of RNA. There are three types of RNA. mRNA or messenger RNA. What is the function of mRNA? It provides a template for translation. So it is a template for protein synthesis to take place. We have learned that DNA cannot directly synthesize proteins. It is the function of the mRNA to synthesize proteins. The next type of RNA is the ribosomal RNA, that is rRNA. It is having 12 functions: structural and catalytic role. What is the structural role? It forms the structure of the ribosomes. And catalytic role In the case of bacteria, it is acting as the enzyme ribozyme. It's having a catalytic function. And tRNA, that is transfer RNA, it brings amino acids for protein synthesis and reads the genetic code. So the three types of RNA are mRNA, rRNA and tRNA, which are having specific functions. Francis Crick postulated the presence of an adapter molecule. And what is this adapter? It is a device which connects two equipments together which cannot be directly connected. So an adapter helps to connect two equipments together. In the same way, Francis Crick postulated the presence of an adapter molecule, and this adapter molecule can read the code and to link with the amino acids. So it is having dual functions. One, it can read the code, and second, it can link to an amino acid. So tRNA or transfer RNA is called as an adapter molecule. Why? Because in the structure of the tRNA, we can see it is having three loops. Okay, three loops, and this is an additional fourth loop. And this loop is called as an anticodon loop. This anticodon loop, it is having a specific function. It is having the basis which is complementary to the codon on the mRNA. So the anticodon loop, it is having the basis which is complementary to the codons which is present on mRNA. It is having an amino acid acceptor end at the 3' end. And it is also having a ribosome binding loop. And also, in addition, it is having an enzyme binding loop. Thus, it is acting as an adapter because it is having the ability to read the codon on the mRNA and also to link to a specific amino acid based on the basis which is complementary to the codons present in the anticodon loop. For initiation, there is a specific tRNA, and this is called as an initiator tRNA. So there is an initiator tRNA which can initiate the process of translation, and there is no tRNAs for stop codons. And the secondary, the 2D structure of tRNA looks like a clover leaf model. This is a clover leaf model which is a 2D model. Whereas the 3D structure of tRNA is an inverted L structure. It is an inverted L structure. Dear children, in this module we learned about mutations which can happen in the genetic code, and also we learned about the three types of RNA. Thank you.